Hi everyone, Professor Gustin here. We're in the last of our 4 videos for prokaryotic cells. In one of the learning outcome we're going to look at right now says describe the structure and function of bacterial chromosomes and plasmids and recognize them on diagrams. So remember, most of the cell information that you know is about eukaryotic cells, and eukaryotic cells have a nucleus and other membrane-bound organelles. Bacterial cells or prokaryotes do not have a nucleus, but they do have a chromosome. So in eukaryotic cells, particularly human eukaryotic cells, we have 46 chromosomes, 46 pieces of DNA in the nucleus of the cell. In the case of prokaryotes, the bacterial cells, they don't have a nucleus, but they do have a chromosome. Now remember, bacterial cells are substantially smaller than eukaryotic cells. Hi everyone, Professor Gustin here, and we're in the last of our 4 videos on prokaryotic cells. Our first learning outcome says describe the structure and function of bacterial chromosomes and plasmids and recognize them on diagrams. So I just want to back up and remind you what you already probably know about chromosomes. Remember eukaryotic cells, cells like our own cells, have a nucleus, and inside that nucleus for humans in particular, are 46 pieces of DNA or 46 chromosomes. Remember, our DNA contains the information on how to make all the parts of the cells. It's our genetic code. So bacterial cells or prokaryotic cells, being substantially smaller than eukaryotic cells and not quite as complex in what they do compared to most eukaryotic cells, they don't need quite as much DNA. And they don't have a nucleus. So in bacterial cells, the DNA is in the form of one chromosome, and that one chromosome is one circular piece of DNA. So if you look at this cartoon, you'll see they have this term nucleoid. Well, bacteria or prokaryotic cells don't have a nucleus. They don't have a membrane around that. But when you look at them under like a transmission electron microscope, you kind of see this lighter area right here. And you're like, what the heck is that? Is that a nucleus? And remember, prokaryotic cells do not have a nucleus, but they have a chromosome. And the chromosome doesn't stain the same way as the rest of the cytoplasm. So a lot of times you can see this, and so scientists gave that a name. They called that the nucleoid, and it just means the region where the chromosome's found, or you hear it called the nuclear region. So if you look at the nucleoid, what you will see is the bacterial chromosome. So like I said, a bacterial chromosome is a single circular chromosome. If you take this big jumbled piece of DNA and you shook it out, it would be one circular chromosome. So remember, humans have 46 linear chromosomes. Most bacteria have one single circular chromosome. And because that chromosome is relatively large compared to the cell, it's all kind of folded up and packed into that cell. Okay. The other genetic material in many prokaryotic cells, not all, but in many cells, are pieces of DNA that we call plasmids. And you can also see the plasmid on this diagram is drawn as a circle. And you can have one plasmid or you can have hundreds of plasmids in a cell. Plasmids are also made of DNA, and you can see they're circular pieces of DNA. They're much smaller than the bacterial chromosome. And remember, the idea with DNA that you learned in another class is that DNA is the instructions on how to make all the proteins in the cell. The DNA is used to make RNA, and RNA is used to make proteins. The units on the DNA are called genes. One gene is going to make a protein. It has enough instructions in it to make a single protein. Well, plasmids, obviously because they're smaller, are going to have a lot fewer genes. They're going to have a lot less instructions on how to make chromosomes, how to make parts of bacterial cells. The chromosome is going to have a lot more a lot more instructions, a lot more genes. And it turns out that the bacterial cells that have plasmids don't need them for routine activities, for normal cell functions. All the genes for normal cell functions are going to be encoded on this chromosome. But plasmids can have genes that give the bacterial cell special functions. One of the topics we'll talk about this semester is antibiotic resistance. How can a bacterial cell that's sensitive to an antibiotic, that's easily killed by an antibiotic, suddenly acquire the ability to be insensitive to the antibiotic? And we call that antibiotic resistance. Well, sometimes bacterial cells acquire a plasmid, and the plasmid has genes on it that make them antibiotic resistant. So it gives the bacterial cell a superpower. There are plasmids that make bacterial cells resistant to exposure to heavy metals. And that's a superpower that allows this bacteria cell to be able to grow in an environment that's got more heavy metals than the typical bacterial cell could handle. So plasmids are not essential for normal life, but carry genes that give bacterial cells kind of special powers. The next learning outcome is describe the structure and function of bacterial ribosomes and compare prokaryotic ribosomes to eukaryotic ribosomes. This is another topic that you probably remember from a previous class. See if you remember what ribosomes do. Ribosomes are, in this picture, they're all these little dots. In your eukaryotic cell picture, there's lots of ribosomes in there. You might remember some of them are on the endoplasmic reticulum, and it's called rough ER when they have lots of ribosomes. But when you look at this cell, you're like, ooh, there's lots and lots of ribosomes, even in a small bacterial cell. So they must be pretty important. Do you remember the function? Ribosomes are the site of protein synthesis. Remember, proteins are the molecules that make up the enzymes, that make up a lot of the structure of cells. They're really important. So ribosomes are where the proteins are made. And so bacteria have a lot of ribosomes floating around in the cytoplasm, just like eukaryotic cells do. And so when we talk about prokaryotic cells not having membrane-bound organelles. Ribosomes aren't actually made of membranes. They're made of protein and RNA, actually. But so they're the one kind of cellular structure that's similar in eukaryotic cells and bacterial cells and prokaryotic cells. So the ribosomes, this kind of organelle that they have. So I wanted to mention that both in eukaryotes and prokaryotes, ribosomes are essential for making proteins. But there is a big difference that I like to mention. We're going to talk again later on in the semester about antibiotics. And it turns out that some antibiotics like streptomycin and tetracycline actually attack ribosomes. So if they can stick to the ribosomes, they stop protein synthesis and they will kill the bacterial cell. But it turns out that these antibiotics don't stick to our own ribosomes. And that's because eukaryotic ribosomes, like ours, and prokaryotic ribosomes, like those in bacteria, are kind of different sizes. They're made up a little bit different way. And this difference allows them to be a good target for antibiotics. Again, we'll talk about this more as the semester goes on, but for a drug to kill a bacterium but not kill the host, there has to be something unique about the structure it attaches to. So ribosomes are good targets for destruction by antibiotics in bacteria because eukaryotic ribosomes, our own ribosomes, are larger than prokaryotic ribosomes and are not sensitive to being attacked by tetracycline and streptomycin and some other antibiotics. While we're on that idea, another part of the cell we talked about, the cell wall that has peptidoglycan in it. Humans don't have peptidoglycan. We don't have it anywhere in our body. So the drug penicillin, the antibiotic penicillin, sticks to peptidoglycan. So it can kill some bacteria that have peptidoglycan, but it won't harm us. So when you're looking for drugs, you're looking for something unique in the bacterial cell that will prevent it from, unique from the host, that will kill the bacteria but not kill the host. The last topic I have in this whole lecture series is second to last topic. Describe the appearance and function of inclusions. Now, if you look at some of these cells, look at this one in particular. These are nice spiral-shaped cells, but they got these little white dots in them. And a lot of times students will look at that under the microscope and they'll say, oh, it looks like there's some organelles or something in there. But remember, prokaryotic cells don't have organelles. These things that didn't stain quite as neatly, as smoothly as the rest of the cell, are called inclusions. An inclusion is a deposit of some kind of chemical in the cell. And they don't, a lot of times they don't stain as homogeneously as the rest of the cell. So inclusions are some kind of chemical. Bacteria can store all sorts of chemicals for all different reasons. In this bacteria, this is some kind of sulfur-containing compound that this bacteria seems to need a lot of, so it stores it up. I'll show you another picture. One of the main reasons why I even mention inclusions is in a live class, people will be looking at their bacteria and they'll say, like, what is that? You know, that light spot in there or something like that. And I don't know what it is. It's just something that's not staining well. I'll say, oh, it's some kind of inclusion. I'll find my picture. I wanted to show you. Oh, I lost it. Yes. There we go. I wanted to show you this bacteria. Oh, this one right here. When we looked at this picture, I look at these bacterial cells and I said, these are bacillus-shaped bacterial cells. But look at, they've got, you know, they kind of look like they're dots. They're not staining evenly. And I would say there's some kind of inclusion in there. There's some kind of chemical deposit that bacteria is storing up. So it's not staining evenly. Here's the same ones I showed you on the other page. There's some inclusions in there. They're not staining very well. Where those are kind of small, but these don't have any inclusions in them. They're staining really evenly. So I just saved that word just in case. And here's some more highly magnified pictures of cells with inclusions, some kind of storage chemicals. And this kind of bacteria actually stores iron in it, and it becomes, it responds to it becomes magnetic, and it aligns itself with the magnetic fields on Earth, believe it or not. Here's the last topic I want to talk to you about. Last learning outcome, explain the function of endospores and what causes sporulation and germination. Endospores are one of those things that affect your life every day, whether you know it or not. And just like when I said biofilms, you know, biofilms, the way we brush our teeth, the way we wash our dishes, a lot of the techniques we use is to remove biofilms. Well, a lot of the food processing techniques we use to store foods and to make food safe is directed to endospores. So what the heck are endospores? First of all, I want to tell you that most bacteria do not make endospores. Endospores are special, and only a couple kinds of bacteria make them. What an endospore is, it's a structure made by some bacteria that allows them to survive in extreme environmental conditions. So it's the kind of thing like if you had, I don't know, a bomb shelter. You could, you know, you're a regular old person, all of a sudden you're like getting worried there's going to be a bomb. You go in your bomb shelter and you have this place where you're protected until things get better, and then you can leave. And so the endospore is kind of like that, although the bacteria itself, the whole bacterial cell kind of degenerates, and it just becomes an endospore, this resting structure. So endospores are really tough. Endospores can survive very high temperatures. They can survive lack of food, lack of water, exposure to radiation, much better than other cells can. I just wanted to see if I put a word on here. And so what the endospore does, this picture looks like what we do in the live lab, and you guys will do this in a virtual lab. The red cells, this is called an endospore stain. The red cells are what we call normal bacterial cells, which I'm going to call vegetative cells. When bacteria are growing and actively living, we say they're vegetative. That's a good thing. They're growing, living, doing their normal stuff. But when they get stressed, these cells that generate endospore, that can make endospores, will do so. And you can see these little green dots inside some of these vegetative cells. So these endospores are forming inside the cell. Something happened to this culture. They were running out of water, it got too hot, they were running out of food, those kind of things. And they got a signal to say, make an endospore because you need to stop. So you can see the endospores are forming inside the cells, but there's also some outside the cells. So eventually the whole vegetative cell, the living, growing cell, will just die off and leave only the endospore. So, for some bacteria, endospores have been isolated that are like 1,000 years old. They haven't been growing for 1,000 years. So some of these endospores are incredibly stable. The reason why we can't just heat up our food all the time and kill bacteria, most bacteria die if you just heat it up on the stove, but we actually have to boil it and boil it for like, did you ever can food? You want to totally eliminate all bacteria, you have to pressure cook it. You have to, or I have to put things in an autoclave to actually inactivate endospores. So a lot of the ways that we process our foods, like canning at very high temperatures, is there to kill endospores. So a couple of vocabulary words related to endospore. Sporulation happens when a vegetative cell, living, growing cell, suffers some kind of bad environmental conditions and starts to make a spore. And that's what it's showing you down here. This is a regular bacterial cell that was under stress. So what does it do? It copies its DNA. It starts surrounding that new piece of DNA with membranes and all sorts of peptidoglycan, a really protective layer. And ultimately, an endospore forms inside of it. The vegetative cell disintegrates, and you just have an endospore. Okay, and this is just a cartoon showing you, here's the red is a vegetative cell. The red is the vegetative cell with an endospore forming in it, and the green is, these are just the free endospores. The endospores are important in health and wellness. There's all sorts of bacteria that make endospores that can cause really serious diseases. So I say, name the two most common genera of bacteria that make endospores and some of the medical implications. A genera, plural for genus. So the two genera that I have that make endospores are Clostridium and Bacillus. Now, fairly recently, they've renamed Clostridium to Clostridioides. So you might see Clostridioides or you might see Clostridium. But either way, the genus begins with a C, so we call it C and whatever species. Okay, so you want to remember these names, Clostridium and Bacillus. These will come up again. I want to show you this. If you take bacteria and you just stain it with regular, just a regular simple stain, you can see if they're making an endospore, the endospores aren't going to stain as well. So I can even see the endospores in there without doing an endospore stain. And in these, you can see the endospores and the vegetative cells, green endospores inside the red vegetative cells. And so these bacteria are most likely in the genus Clostridium or Bacillus. I'm going to see if you can guess any of these diseases. This one, it says, here's a soldier dying of tetanus. This is a kind of a classic painting. And here we go. I've got a bunch of names here. Clostridium tetani is the bacterium that causes tetanus. And Clostridium tetani make a toxin that causes the muscles to contract. And so that's what happens when you get tetanus. You get lockjaw, you can't move your jaw, all your muscles get contracted, and you can actually contract your muscles to the point that you break your ribs, you break bones, and you stop breathing. Another one that you probably heard of is Clostridium difficile. For those of you that work in healthcare, especially in nursing homes, you might hear of patients getting C. diff. It's a really bad form of diarrhea, and usually most common in older people. That's why it happens in nursing homes, especially if they've been taking antibiotics. So C. diff is a bacteria that makes endospores. So you got a patient that has Clostridium difficile. That bacteria in those feces can stay around for years if you don't get them out of your environment. So this is something that's just, these things are around, they're hard to kill because they make endospores. We've all heard of botulism. Clostridium botulinum is the bacteria that causes botulism, and it also makes a toxin, but unlike Clostridium tetani, botulism causes flaccid paralysis. So if you have tetanus, all your muscles contract. If you have botulism, all your muscles relax to the point, again, that you can't breathe. And so in this picture, this child can't even open their eyes. They have botulism. And both of these diseases are very deadly. The last one that you've probably heard of, but you might not have heard the name. You've heard of the disease. You've probably heard of people getting gangrene. You have tissue damage. And a lot of times people that have diabetes, their blood vessels are kind of not working as well, especially in their extremities. They don't get enough blood to circulate. They can get infections, and they can get gas gangrene, which is an infection of Clostridium perfringens. Once your tissue does this, it's completely dead, and needs to be amputated. It's not going to revive. So it smells really bad because the bacteria smell really bad. And this is a bacterium that's around. It can be in the dust. It can be just on our bodies. And if it gets into a wound, it can cause this disease. So all these endospore formers can be very, very serious. The last one I wanted to mention, I didn't write it down here, but the genus Bacillus, you've probably heard of anthrax. And anthrax is a disease caused by Bacillus anthracis. So even the bacteria in the genus Bacillus can cause diseases. Bacillus cereus is a foodborne pathogen, and that's a endospore former. So there we go. You're going to talk a little bit more about endospores in lab, and that's the end of this unit.